I'm going to walk you through some of the blood basics in this video, including the different components of blood, the different blood types, and even how some of the blood tests work. Before we totally jump into blood, we're going to talk about serology real quick. Serology is the examination and analysis of different bodily fluids. So this would include blood, but it also includes urine, sweat, semen, saliva, so on. All right, so anytime you hear the word serology, it's talking about the analysis here of bodily fluids, including blood. So now I'm going to walk you through the different components of blood. There are four different components that we're going to look at here. The first is the red blood cell. Okay, red blood cells are also known as erythrocytes. They have the very important protein called hemoglobin. Hemoglobin will attach to the oxygen and bring it to different cells in the body. So the red blood cells are produced in the bone marrow, and they are the most abundant cells in our body. So if you look at the picture here, okay, these red blood cells, I like to think of them as being like a donut without the hole being fully punched through. All right. The second component of blood is going to be the white blood cells, also known as leukocytes. They're part of your immune response, and they're going to be responsible here for destroying infectious agents. They're going to be secreting antibodies. So if you look at the picture over here, all right, they kind of look like a, fluffy cotton ball. So those are the first two components of blood. Then we have the platelets. Platelets are also known as thrombocytes, and these are the clotting factors that are in the blood. These are important for, if you get a cut and, you want to obviously stop bleeding, those platelets help seal the wound and make the blood clot so it stops continuing to flow. So if you look at the picture here, you can see the arrow. They are very kind of small, all right, but they are included in the blood, very important function. And then we have the plasma. The plasma is the yellowish liquid portion of the blood. This is where all your nutrients, vitamins are found, and it does make up about 55% of the blood. All right, so those are the four different components of the blood that you need to know. So again here, real quick, if you want to look at this in picture form, you can see the red blood cell kind of has that circular shape, looks like a donut without the hole fully punched through. The fluffy cotton balls are the white blood cells, and then you have the platelets here, which are smaller, and then the liquid portion is the plasma. I just wanted you to see some different microscopic views here of different kinds of blood. So you can see a couple different animals here, cat, snake, frog, and then you can see how it compares to humans. So you definitely see some similarities and some differences, but it's definitely neat to see some of these different views here. There's some different blood facts on this slide. So an average adult person has about 5 liters of blood, which makes up for about 7 to 8% of a person's body weight. So in theory, if you drained all the blood out of a person, they would lose about 7 to 8% of their weight. Blood is considered a living tissue. It is carrying oxygen and nutrients to all different parts of the body, but it's also taking those waste products out. So it's bringing the carbon dioxide to the lungs, taking other waste products to the kidneys, so on. Blood is also important here in fighting infection. All right, we want it to be, sealing our wounds. We want it to be part of our immune response so we can stay healthy. And there are 1 billion red blood cells in 2 to 3 drops of blood. Okay, so that's just trying to give you an idea here of how many red blood cells there would be in just 2 to 3 drops of blood. For comparison, for every 600 red blood cells, there's only about 40 platelets and 1 white blood cell. Okay, so the white blood cells are not as common as the red blood cells. Now I want to talk about the different blood types here. So your blood type is determined by your genes before birth, and it depends on specific proteins that are present on your red blood cells. We call these proteins antigens. Okay, so your blood cells have specific proteins on them, or maybe they don't have any proteins at all if you have type O blood. All right, but there are these proteins on the red blood cells that we call antigens. Okay, they are stimulating an immune response. They're also known as agglutinogens. All right, And then we have these antibodies, which are these Y-shaped proteins, which are found in the blood, and they're used by the immune system to recognize foreign invaders here. Basically, the antibodies will bind to a specific antigen. They bind to a specific protein on another cell. All right. So if you look here in the picture on the right, okay, if you are type A blood, your red blood cells are going to have the A antigen, the A protein essentially, all over the red blood cell. If you had type B blood, your red blood cells are going to be covered with the B antigen. If you have type AB blood, you're going to have both A and B antigens all over your red blood cells. And if you're type O blood, you're not going to have any of those antigens on your red blood cells. All right. But then there are certain antibodies that you're going to produce that will bind to these different antigens. And we'll talk more about this. So your body has this immune response, okay? Anytime a foreign invader comes into your body, this leads to the creation of these antibodies, all right? These antibodies will recognize that invader. So if the invader comes back into your body, these antibodies will kind of attach to it, cause it to kind of clump up so that it can be destroyed. All right. So if you look at this in the picture, you can kind of see that here. These antibodies attach to specific antigens, right? Specific proteins on the cell, and it causes the clumping up. You can even see it taking place here in the picture on the right. Right. This clumping is called agglutination. Basically, the antibodies recognize a specific antigen or a specific protein. All right. And this causes clumping. So obviously, if this starts to happen with your blood, that would be a problem. All right, and we're going to talk more about this with blood types here as we go forward. As I kind of mentioned earlier here, your blood type is determined by which antigens, which proteins are present or not present on your red blood cells. Okay, so if you are a person who has type A blood, okay, you are going to have A antigens all over your red blood cells. All right, if you have type B blood, you're going to have B antigens all over your red blood cells. Okay, but then you produce antibodies that are kind of opposite to that. So if you have type A blood, your body is going to produce B antibodies, okay? Basically, you would not produce A antibodies because they would attack the A antigen. And you have A antigens all over your red blood cells. So it produces the opposite antibody. If you have type B blood, you would produce A antibodies. Basically, those A antibodies are looking for any foreign antigens. A antigens are foreign to your body because you only have B antigens on your red blood cells. If you have type AB blood, you have both A and B antigens. Therefore, your body can't produce A or B antibodies. You would have no antibodies being produced because your body recognizes both A and B antigens. But if you're type O blood, notice you have no A or B antigens present on your red blood cells. Okay? Therefore, since your body doesn't have those antigens, you're going to produce both A and B antibodies because your body does not recognize them. All right? If an A or B type blood entered your body here, you're going to essentially be attacking it because you produce both A and B antibodies. So you have to think about it in terms of whatever type you are, that's the antigens you see on the red blood cell, and you produce the opposite antibodies that would be looking to attack um, that specific antigen. Okay? So you really got to think about this here. There's something else here called the rhesus factor. This is another protein that is found on the red blood cells of some people, but not others. Okay, so scientists studied these rhesus monkeys and they discovered this protein on the cells, and they noticed it's found in some people and not found in other people. So they call this the Rh factor. So if your blood has this protein, all right, you would be considered to be Rh positive. So if you might have heard of like, oh, somebody has A positive blood, that means they have type A blood, but they also have that Rh factor present on their red blood cells. If you don't have that Rh protein, you're going to be considered Rh negative or type negative blood. Okay, so this is just another protein that's found on these red blood cells in some people and not in others. This is an interesting picture here. I just want to kind of point this out to you. So if you had an Rh negative woman, so a woman who does not have the rhesus factor on her blood cells, having a baby with an Rh positive man, all right? So let's just say that the fetus here now, Rh positive, all right? So what happens is as the um, fetus is growing, eventually she gives birth, some of these cells from the Rh positive fetus enter her bloodstream, and now she is producing some of these um, antibodies, all right? So she now has antibodies to Rh positive, all right? So what happens here is in the next pregnancy, if she has another Rh positive baby, she has antibodies present in her body which could attack that fetus. All right, so this is just something interesting. Just wanted to get you thinking about this and point that out to you. So obviously, blood transfusions are when you need to be given blood for whatever reason, due to surgery, illness, injuries. So you, as we talked about here, you have to be careful about which type of blood you get because your body is producing antibodies here that might attack a specific type of blood. So the universal donor is type O blood. Okay, type O blood has no antigens, no A or B antigens on the surface. So if O blood is given to a person, all right, there's no antigens to be attacked. Okay, no matter what antibodies you produce, there's nothing on that O blood cell that can be attacked by the antibodies because it doesn't have any antigens. And then AB is considered the universal acceptor, okay? Because type AB blood produces no A or B antibodies, all right? So no matter what blood a person whose AB is given, okay, they don't have antibodies that's going to attack either A or B antigens, all right? However, here, if you are Rh positive, you can get positive or negative. But if you're Rh negative, you can only receive type negative blood. All right. So this picture over here kind of does summarize this. You can see that O can give to everybody and AB can accept from everybody. This table is just showing you the different types of blood and the antigens present and the antibodies produced and which transfusions would be safe here. All right. So obviously, you have to be careful with this. All right. So we'll just go through one of them and then you can look at the table. But remember, if you're type A blood, you have the A antigen, the A protein on your red blood cells. So therefore, your body is producing B antibodies. Your body is not going to recognize B antigens. So you're going to have B antibodies present. All right. So a person with type A blood can give to another person who's type A. You can always give to the exact same type, but it can also give to a person who's AB. Because remember, AB can accept from everyone. All right. But if you have A blood, you can only get blood from another person who's type A or O. All right. You can't get B blood because you produce B antibodies. You can't get AB blood because, again, AB blood would have the B antigen present on it. You would have B antibodies and you would attack it. All right. Basically, the blood would agglutinate here. You would see clumping if you got a type that you cannot get. All right. So definitely look this table over. Make sure you understand this idea that you can only um, give to certain people and receive certain types of blood here. This slide is just showing you how common the different blood types are. So if you're type O blood, all right, that's about 46% of all people, okay? And then the lowest type of blood here is AB. Only about 4% of people have type AB blood, all right? And you can even see the differences here from, say, O positive, O negative, and AB positive, AB negative. You can see the negatives are even usually less common. All right, so this is just kind of more for your information. Now that we went through some of the, blood basics and, the different components of blood, we want to see how this can actually be useful here in forensics. So a lot of times, blood is found at a crime scene. All right. So there's some different things you need to answer here about the blood. First, you have to find out, is it blood? All right. You have to confirm that it actually is blood. Then you got to figure out if it's, animal or human blood. Then, identifying possibly whose blood it is, what type it is. You might be able to analyze the blood to see if there was, alcohol or drugs present. And then obviously you want to figure out, how did it get there? Okay, so that's when you would analyze like blood spatter evidence. All right, so there's a lot of information you want to kind of figure out here from the blood when it's found at a crime scene. There's different ways blood can be used as evidence. So if you have blood samples here, you obtain these samples, you can analyze them, determine the blood type, you can even get DNA from that. All right. If you find blood droplets at a crime scene, this could give you information about, the location of the crime and the victim. You can figure out maybe if the victim was moving around, maybe even figure out the type of weapon. And then there's something even called blood spatter here, which we're going to look at in a different video more in depth. Basically, blood spatter is like the patterns can be analyzed to figure out what happened at a crime scene as well. All right. If you're just looking at blood and determining the type, that's going to be a form of class evidence. All right. Because that does not point to a specific person. Unless you look at the DNA in the blood, if it's present, if you can get enough in the sample, uh, it's not going to be individual evidence unless you get the DNA. If all you do is determine the type, that's going to be a form of class evidence. The last thing I want to talk about in this video are these blood typing tests. These are very important tests, something we can look at even in lab. But you need to realize here, DNA is great. Being able to identify the exact person is great. But sometimes, doing a blood typing test is going to be much faster and way less expensive here. So if you could do a blood typing test and realize, wow, the person we thought was the suspect doesn't have the type of blood that we are looking for, then you can kind of save a lot of money here and not go any further. All right. So to do these tests, you add these specific anti-serums, which will clump with specific types of blood. Essentially, these anti-serums clump when they sense specific antigens are present. Okay. So for example, if you added what's called anti-serum A or anti-A to a sample of blood and it clumps up, you know that the A antigen is present, okay? So that could be that it's type A blood. If you add anti-B to a sample of blood and it clumps, that tells you then B is present, okay? So if you take a sample of blood and add anti-A and anti-B to different, samples of it, and they both clump, that tells you both A and B antigens are present. So it would be type AB blood. If you added both A, anti-A, and anti-B to a sample of blood, different, samples of the same blood, and neither of them clump, well, that tells you neither A or B is present, so it would be type O blood. So if you want to look at here at the picture, all right, clumping kind of just, you literally start to see like the blood kind of clump up a little bit. All right, over here on the right, there's no clumping here. So these blood typing tests are usually set up in 3 different little wells. All right, I don't know if you can notice here, but the top left, there's a little A there. That's where they're going to add anti-A to. Down here on the bottom, this says B. That's where they would add anti-B. And over here on the right, that's where the RH, where it's going to be either positive or negative. All right, so if you look at this sample, it's labeled as B plus because if you look, when they added anti-A to the sample of blood, it did not clump. So there's no antigens present. But you see the clumping when you added anti-B. There's clumping here in B, all right? And there's clumping in the RH. So that tells you it's positive for the RH. If you see clumping in the RH, it's positive for the RH factor. So this would be type B plus blood because it clumped when you added anti-B. So B must be present, and it clumped when you added the RH, which means the RH is positive. All right. Down here, this is listed as O positive. All right. If you look for A and B, there was no clumping when you added those antiserums. So A and B are both not present, like you would expect for type O blood. But you do see the clumping present for the Rh factor. So that means it's got to be positive. All right. So these blood typing tests are important. Make sure you understand this idea that if you add anti-A and it clumps, then A antigens are present. And if you add anti-B and it clumps, then B antigens are present. All right. So make sure you understand this.